All right, guys, chapter 15 is probably my favorite chapter because it really puts together everything that we've been talking about with cell division and genetics and cell biology. We've been talking about these different things. We already have the word for chromosomes, right? So today we know that Mendel's factors were actually stretches of nucleotides along the chromosomes that we can see under the cell. So this image here is actually of a single-celled C. elegans embryo. The DNA is actually all in red, but genes that are on have been labeled in green. And there is a special fluorescence tag here called fluorescence in situ hybridization that's marking a particular gene that is on this chromosome where the whole rest of the chromosome is off. The fact that those squiggly little lines that scientists could see under the microscope doing the steps of mitosis and meiosis, and the fact that Mendel's factors for heredity were the same thing wasn't put together until the experiments we're going to talk about today by the Morgan Hunt group. I highly encourage you guys to go check out the link that I provided with this chapter to the citable article where it talks about all those aspects and the timing of the cell biology and the genetics and the experiments with fruit flies that came together to give us our modern view of inheritance by genes on chromosomes. So we've already been assuming this as we've gone along. The fact that you have two of every chromosome, one from your mom and one from your dad, explains the law of segregation, right? Those two different homologous chromosomes separate from one another during meiosis. And the fact that you can get F2s that look different than either the P generation or the F1 generation is based on the fact that genes on different chromosomes can get inherited in different combinations, or the law of independent assortment. On not homologous chromosomes, so the long chromosome versus the short chromosome, can be passed out in different combinations than they were inherited by that individual. These two laws together take parents that are heterozygous at two different loci on two different chromosomes and can go through meiosis to produce four unique gametes that have different combinations, either dominant with dominant, recessive with recessive, or one dominant with one recessive, and one recessive with one dominant. But the understanding for how this happens didn't come together until the Thomas Hunt Morgan experiments. They were using fruit flies as a model organism. Fruit flies are useful for model organisms for many different reasons. First of all, nobody really cares if you smash a couple of house flies, so you don't have as many ethical constraints as you would if you were working with vertebrates. Second of all, they have a very large brood size. They have many offspring at a time, and they can go from generation to generation very quickly. It only takes 2 weeks to go from egg to sexually mature adult. Also, they make good genetic model organisms because they have only 4 pairs of chromosomes. Doing genetics in humans, where we have 23 different pairs of chromosomes, is a lot more complicated. So the first experiments were using fruit flies as model organisms. So the terminology for the traits that we're going to look at in fruit flies are a little bit different. When we see a common trait, okay, that's called the wild type trait. And the wild type trait is usually the dominant trait. So in fruit flies, the wild type trait for eye color was red, but there were mutants that had white eyes. And so the mutant phenotype is usually the recessive phenotype. The other confusing thing is the way that the geneticists named the genes was after the novel unique phenotype. So the gene that controls eye color in fruit flies is called the white gene. Even though normal flies have red eyes, if you're homozygous recessive for the mutation, you have white eyes. So the eye color gene is called white. So once some of these mutants were identified, they wanted to set up the same type of crosses that Mendel had set up with his pea plants. So they wanted true breeding parents with red eyes and true breeding parents with white eyes. And then once they had stable populations established, then they could set up crosses. They had these true breeding populations established. They set up the classic cross. So they had white-eyed flies that were true breeding, which they assumed were homozygous, crossed to red-eyed flies that were true breeding, that were homozygous. And in the F1s, all of the offspring had red eyes, both males and females. And so they expected that in the next generation, if you cross heterozygous parents, you would get a 3-to-1 phenotypic ratio, where 3 out of 4 would have red eyes and 1 out of 4 would have white eyes. And that was true, that's what they got, but they noticed something strange about those F2s. All of the females had red eyes, but only males had white eyes. Okay? So they got the 3-to-1 ratio, but it was only males that had white eyes. And that made the researchers say, aha, I think I know what's going on here, because of the way that sex is determined in fruit flies. In fruit flies, just like in humans, females have 2 of the same sex chromosome, and males have 2 different sex chromosomes. In organisms where this is the case, the chromosomes that are the same in females are called the X chromosome, and the chromosome that's unique to males is called the Y chromosome. So for both humans and fruit flies, XX is female and XY is male. Now, the X chromosome in humans is not homologous to the X chromosome in fruit flies. They have totally different genes on them. Okay. In fruit flies, the gene for eye color was actually on that Y chromosome. So we can redraw that Punnett square in a different way. Those P0 males with the white eyes had 1 copy of the recessive mutant white eye gene, but the Y chromosome doesn't have any of the same genes. It has no genes for eye color on it. The females who had the red eyes were homozygous dominant for the red eye color gene. So when we look at the F1s, all of the females got a dominant copy of the allele from their mom and a recessive copy of the allele from their dad. The females truly were heterozygotes. But the males all got a white copy of the dominant red copy of the gene from their mom and no copy of the gene for eye color from their dad. Now, whatever 1 copy of the gene they had, that's the phenotype they had. So even though they had 1 copy, they still had the dominant trait, the red eye color. Now, when we cross these guys to make the F2, something different happens. Females were truly heterozygous. The males we describe as being hemizygous. They have 1 copy of the gene that we're interested in. And their other chromosome was the Y chromosome that has totally different genes. So all of the daughters got an X chromosome with the dominant red eye allele from their dad. All of the sons got the Y chromosome with no genes for eye color from their dad. Half of the offspring got the dominant red eye gene from their mom and had red eyes. And half of the offspring got the recessive white eye allele from their mom. These daughters are heterozygous and have red eyes, but these sons are hemizygous. They have just 1 copy of the gene, and the 1 copy they have is the recessive white allele. So they have the white eye phenotype. So for the F2, we have the same genotypic ratios that we would expect from a heterozygous cross. We have 1 out of 4 being homozygous dominant. These 2 are heterozygous kind of in different ways. These females are really, truly heterozygous, whereas these males have 1 functional dominant copy, and then the other chromosome is not homologous. It has none of the same genes, and they have the red eye phenotype. And then these males down here have just 1 copy of the recessive allele and have the recessive phenotype. So we get the same phenotypic ratio. 3 out of 4 have the red eye phenotype, 1 out of 4 have the white eye phenotype, but only the males have white eyes because the gene for eye color is on the Y chromosome. A couple other things about sex chromosomes before we go on. So first of all, in organisms where females have 2 of the same sex chromosomes and males have 2 different sex chromosomes, we describe them as females are XX and males are XY. However, like I said, the X chromosome in humans is not homologous to the X chromosome in fruit flies. They don't have the same genes on them. So in humans, the genes on the sex chromosomes determine sex determination. Importantly, there's a gene on the Y chromosome called SRY, or sex regulation on the Y, which initiates male sexual development. You have to have a copy of the Y chromosome to develop into a male, unless you have some mutation like translocation that moves the SRY gene from the Y onto some other chromosome. So the default setting for humans is actually female, and it's not until embryos get this positive signal from the SRY gene on the Y chromosome that male sexual development begins. So if you have a mutation in the SRY gene that makes it non-functional, you could be an XY female because you never initiate male sexual regulation. Also, if you have just one X chromosome and no other sex chromosome, so your XO, those individuals also develop as pretty much normal, healthy females. It's called Turner syndrome, and it's the only chromosomal aneuploid or missing an entire chromosome that's tolerated in humans. So sex determination is actually regulated in different ways in different organisms. In chickens, it's the opposite way. So most birds, it's the males that have two of the same sex chromosome, and it's the females that have two different chromosomes. So in birds, females are the heterogametic sex, the ones that have two different kinds of gametes. And the males all have the same kind of gamete. So to indicate this, in organisms where the males have two of the same sex chromosomes, they're called ZZ, and females that have two different chromosomes are called ZW. But there's other strategies. So some insects determine sex by the number of sex chromosomes they get. So in some insects, having two sex chromosomes makes you female, whereas having only one makes you male. But in other organisms, like honeybees, it's not the number of sex chromosomes you have, but rather the number of copies of the whole genome you have. So in honeybees, females are diploid. They have two of every chromosome, whereas males are only haploid. They have one of each chromosome. There's other systems too. Some organisms determine sex based on the temperature of incubation of the eggs or the predominance of males and females in the population. So the way that sex determination happens is not universal. What I do want you to be able to do for the exam is do genetics problems with genes that are on the sex chromosomes. Now, normally when we talk about sex-linked genes in humans, we're talking about genes on the X. The X chromosome has thousands of genes on them, all of them important. If you're missing two X chromosomes, you're just not alive. Whereas the genes on the Y chromosome are really exclusively responsible for male fertility and male sex determination, and you don't need them. I get by fine without one every day, and it hasn't hurt me at all. Okay. So usually when we're talking about sex-linked traits, we're talking about genes on the X chromosome for humans. And you can do Punnett squares for humans and fruit flies for genes on the X chromosome. There's actually a limited number of Punnett squares you can do with genes on the sex chromosomes because there's only so many combinations. You can have females that are homozygous dominant, females that are heterozygous, or females that are homozygous recessive. You could have males that have one copy of the dominant gene and a Y chromosome, or males that have one copy of the mutant or recessive gene. So we set up the Punnett square the same way. So we can have, let's say, our heterozygous females, which are carriers. They carry the mutation but are not affected by it, cross to males with a wild-type normal copy of the gene. So the males will give the wild-type dominant functional copy of the gene to all of their daughters, and the Y chromosome with no copies of the gene to their sons. The heterozygous females will give a wild-type functional copy of the gene to half their children, and a mutant recessive copy of the gene to half of their children. So these two are females, these two are males. This female is homozygous dominant or homozygous wild type. This female is heterozygous, is a carrier, but has the dominant normal phenotype. This male has one normal copy of the gene, so has the dominant phenotype, and this male has one mutant recessive copy of the gene and is affected. So you can do these Punnett squares in the same way. Just remember that the Y chromosome has no copies of the gene. So I like to put it in a circle to remind me that there's 0 copies of this gene available. One more example, the mother is heterozygous, the father is affected by the phenotype. The father gives a Y chromosome to all of his sons and a mutant recessive allele to all of his daughters. Half the offspring get a wild-type functional copy of the gene from mom. They have the wild-type normal trait, whereas half the offspring are going to get the mutant allele from mom. These daughters are going to be homozygous recessive, and these sons are going to have the recessive affected phenotype as well. Human traits that are X-linked include colorblindness, Duchenne's muscular dystrophy, and hemophilia. Usually we use colorblindness as an example because it doesn't kill the offspring, so we can keep doing the crosses.